Hi guys. You guys are doing respiratory and digestive in lab this week, so we're going to be misaligned from lab, but then you'll be ahead for next week. I don't generally do respiratory and digestive in lecture and anatomy because it's relatively simple and we need the time for other things. So I'm going to start on urinary system. In some semesters, when things are less crazy, I do lymphatic system, but I'm not going to do that for you guys. So we're going to have urinary system and reproductive system. If it looks like we still have time and energy, which is unlikely given the current climate, then you guys will also have lymphatic at the end. But for right now, I'm kind of planning on your fourth exam being vessels. You already did heart, but vessels, urinary system, and reproductive system. Okay, and again, don't send me an email about it. Just listen to the video. Unless I tell you otherwise, lymphatic and blood will not be on either the fourth test or the final. Okay. So urinary system. So before we get into the anatomy of the urinary system, I want to look at the big picture functionality of the urinary system. So what you have known that the urinary system does since you were little is it removes waste products from the body. And we tend to refer to those as nitrogenous wastes. They include urea and uric acid and ammonia. Other water-soluble waste products are also removed from the body via the urinary system, but the urinary system does this primarily by doing three processes. I'm going to introduce you guys to these, the names of these processes. Do I talk about them down here? Can't remember if I do it. Oh yeah, I'll introduce you to them later then. The filtration, reabsorption, and secretion that you end up using in phys quite often. So I'll introduce you to them in anatomy as well, but I'll wait a couple minutes. Okay, so not only does the kidney, do the kidneys cleanse the water-soluble waste products from your bloodstream, but they're also really, really intimately involved. Oh, shoot, I went down there again. Intimately involved with, hold on just a second. My screen brush back up here. With the cardiovascular system, because whatever goes into the urine came from the bloodstream. And so the more you pee, the lower your blood volume and the lower your blood pressure, the more water you retain at your kidneys, the higher your blood volume, the higher your blood pressure. So they're really intimately involved with regulating blood pressure. And in phys, believe it or not, you talk about them as much with this functionality as you do with the cleansing waste products functionality. So really, really intimately involved with the cardiovascular system. And they also, by sort of deciding what to pee out versus keep in the bloodstream, they are also regulating the water composition of your blood, the ion composition and the pH of your blood by, for instance, deciding how many H+ ions to keep in your bloodstream versus pee out. You can regulate the pH of your bloodstream. And in addition, the kidneys have an extra function that's all involved in the cardiovascular system because the kidneys actually regulate red blood cell production. The kidneys produce a hormone called EPO or erythropoietin. And EPO tells the red bone marrow when to make red blood cells. So basically, big picture understanding is that, yeah, the kidneys get rid of waste products, but they're also completely intimately involved with cardiovascular function. Okay, so now let's just get into the basic anatomy. The big picture anatomy of the urinary system is super duper simple. Basically, of course, we have some organs and we have our kidneys, right? And the kidneys are in the superior lumbar region. One of them slightly lower than the other one. Does anybody remember why one is slightly lower than the other one? So this is your left kidney, this is your right kidney. Anybody remember why your right kidney is slightly lower? the liver is kind of smashing it down there. So, but they're close to the same level, and they are both retroperitoneal. Do you guys remember retroperitoneal from the beginning of the semester? Retroperitoneal means that they were in the body cavity before you put in the parietal peritoneum. So they are behind the peritoneum, retroperitoneum. Let me kind of show you what that looks like in this figure. We'll look at this figure again in a second. This figure is a little hard to understand, but just to show you part of what I'm going to teach you in just a second. All right, so here's the kidneys, right? And this is a transverse or cross section through the L2 region. And what I want to show you is the peritoneum right here. This is the parietal peritoneum, and the kidneys are behind the parietal peritoneum with a couple of layers of fat actually packing them back there. So they are retroperitoneal, and we'll come back and look at this picture in a minute. It's a picture that's actually kind of hard to learn when you're looking at a screen, so I would definitely take this one and stick it into your notes or print it out or whatever you do. Blow it up on your own screen. It's hard to figure out what you're looking at when you're just looking at my screen. Okay, and each of your kidneys is about the size of a bar of soap. And remembering, so if you're looking at them sort of with reference to other things, so here's T12, right, the last vertebra that has a rib attached to it. And so you've got L1 and L2, so you see where they are. This is the last rib, the T12 vertebra right there in L1 and L2. Sitting on the superior surface of your kidneys is the adrenal or suprarenal glands, and you learned those because in the vessels you had to learn the suprarenal arteries and veins that went to and away from those. The adrenal glands are not directly related to kidney function. They make one hormone that's related to kidney function, but they just happen to sit on top of those. And interestingly, they're not generally that big. As far as basic kidney function, if you look at, or basic kidney structure, if you look at this little indentation right here, this is called the renal hilum or hilus. And this is where the vessels, nerves enter, leave, all of the tubes are connected at the hilus or hilum. Hilus or hilum is a really common term for like a central location where everything kind of attaches. So you'll run into one in the ovaries and you'll run into one in a couple of other organs. So it's a really common term, hilus or hilum. And then if we start to look at the protective layers that are around the kidney, we're going to do a cross section through the kidney right here, and we're going to look back at this figure from here. I'm going to blow it up for you so that you can kind of follow along with where we are, and it's a little hard to see. Okay, so let us start at the kidney itself. So if you look at the surface of the kidney itself, the very first layer that you're going to run into is basically the boundary of the kidney, the outer coating of the kidney. It's called the renal capsule or the fibrous capsule. Renal or fibrous capsule. It's on the surface. It's made of dense irregular connective tissue. It's important because it gives a boundary to the kidney, because the urinary tract, pathogens can, for instance, make it all the way up the urinary tract into the kidney, but generally speaking, they're actually sequestered within the kidney because of that boundary that you're running into. I'll draw it on the other one so I don't have to move back and forth. Okay, so the first layer that you run into, which is in that color right there, is called the renal capsule or the fibrous capsule. And then as we start moving out, the next layer you're going to run into is the first layer of fat that is packing around the kidney. And that is called, so here's the renal capsule or the fibrous capsule. And then see this next most superficial layer of fat that's right there. that layer of fat right there, the closest one, is the perinephric fat. Perinephric fat, the fat that is around the kidney. That is also sometimes called the perirenal fat. That's immediately closest to the kidney. And then you're going to run into a layer of connective tissue superficial to that. This is all like packing material around the kidney. A layer of connective tissue superficial to that, which you can see right here, that right there. That is called the renal fascia. And the renal fascia is dense, irregular connective tissue, and it's really kind of smashing the kidneys back against the body wall. I always think about what surrounds the kidneys and keeps them against the body wall, kind of like layers of packing peanuts within layers of packing tape on top. So the kidney was in there, and then this fat is a layer of packing peanuts, and then I've got a layer of packing tape right on top of it. That first layer of packing tape is called the renal fascia, but then I'm going to have another layer of packing peanuts, and that next layer of packing peanuts is right here. That's more superficial, coming from the kidney out, and that's called the perinephric fat. So another layer of packing peanuts, another layer of packing, keeping it back there. And superficial to the perinephric fat is when you are going to run into the peritoneum, and specifically it is the parietal peritoneum. So on the anterior surface, it contacts the parietal peritoneum. So basically what you have is you have kidneys, right? The boundary of the kidneys is the renal capsule or the fibrous capsule. Then I've got a layer of packing material that's called the perinephric fat. Then a layer of packing tape that's called the renal fascia. Then another layer of packing material right here that is called the perinephric fat. And then, especially visible on this side, you're going to run into the last layer of packing tape, which is called the parietal peritoneum or the peritoneum. So all of that is really important for keeping the kidneys smashed to the back body wall, because they're not actually connected via mesentery to the back body wall like the rest of the organs. So that fat is really important for keeping them positioned on the back body wall. All right. So if we start to look at the internal gross anatomy of the kidney, the best way to do it is to look at a coronal section of the kidney. So here's a coronal section of the kidney, and the kidney is one of those organs that when you actually get a wet specimen to look at, it looks something like the model. Of course, nothing's color-coded as beautifully as the models. It just doesn't happen. Did you just stop, mouse? Come here, mouse. Technology is really not liking me today, and the feeling is mutual. Have you ever had that kind of a day? You can do it, mouse. Okay, we'll just go the old-fashioned way for a second. All right. Basically, what we have is, as we move from the outside to the inside in the kidney, we have the cortex. Cortex, if you've got a layered organ like a brain or an ovary or a kidney, the cortex is going to be outside and the medulla is going to be on the inside. So we've got the renal cortex right here all the way around this whole layer, and then we've got the renal medulla on the inside. So cortex outside, medulla inside. The medulla starts where the pyramids start. Okay, so I'm going to show you guys a little animation that just introduces you to the gross anatomy of the kidney. And while I do that, I'll try to get my mouse working. Okay. Get out of there, arrows. Okay, you could just stay there.
The kidneys are large bean-shaped organs located in a retroperitoneal position on the posterior abdominal wall on either side of the vertebral column. The hilum is located along the concave medial border where blood vessels and nerves enter and exit the kidney. It also serves as the point of exit for the renal pelvis, which transports urine to the ureter. The hilum is continuous with an expanded area within the kidney known as the renal sinus. The kidney tissue is divided into an outer renal cortex and an inner renal medulla. Extensions of the cortex, called renal columns, project into the medulla between renal pyramids. The base of each pyramid is located at the junction of the cortex and medulla, the cortical medullary junction. The apex or tip of each pyramid, known as the renal papilla, projects into a funnel-shaped structure in the renal sinus called a minor calyx. Several minor calyces merge to form larger structures called major calyces, with each kidney containing 2 to 3 major calyces. Major calyces merge to form a single funnel-shaped renal pelvis. A human kidney is divided into 8 to 15 lobes, each consisting of a renal pyramid with a renal column on each side, and the cortex external to the pyramid base. Blood is carried to each kidney for filtration through the renal artery. The renal artery branches in the...
Okay, so that'll get us started. We'll come back to the blood in just a minute. So let me get you do the same thing on the figure that they did on the animation, and then we'll come back to the animation. This will help us move through this. Okay, so now let's find the structures that she was describing. Make sure you can find them here. So if we're looking at the internal gross anatomy of a kidney via a coronal section, first thing that we said was that the outer layer right here, from here to here, is the cortex of the kidney. It tends to be lighter in color. It will extend inward the same color as these renal columns. And then the renal medulla is from here to here. It tends to be darker in color. And it is composed of usually 8 to 15 renal pyramids. But this one just happens to show 6. I don't know why they did that. But 1, 2, 3, 4, 5, 6 in this picture. The base of each pyramid faces toward the cortex. The tip of each pyramid faces toward the inner surface of the kidney, this whole region right here, called the renal sinus. The tip of each of these guys, each of the pyramids, and people have trouble understanding this, but the tip of each of the pyramids right here, so it basically goes down and it forms a little tip right there. And the tip is surrounded by this little cup. Well, the tip itself is called the renal papilla, and papilla means nipple. So you got your kidney nipples, you got your heart nipples, you got your nipples everywhere. So the tips of the pyramids, those go toward the medial surface of the kidney. These are called the renal papilla. Each renal papilla is actually surrounded by a cup. So do you see this little cup right here that surrounds the renal papilla? That is actually called a minor calyx. Minor meaning small, and calyx actually means cup. Plural is calyces. So it dips into, each papilla right there dips into a minor calyx. So there is one calyx per pyramid. So the minor calyces, 1, 2, 3, will get together to form a major calyx. And a major calyx receives urine from 3 different minor calyces. And then all of the major calyces will actually dump into the renal pelvis. So the renal pelvis receives urine from all of the pyramids, all of the minor calyces, all of the major calyces. And then the renal pelvis will dump into the ureter, and then the ureter will lead to the urinary bladder. So urine will travel. Urine's going to be made here. We'll talk about how it's made in just a little bit. Urine travels from the renal pyramid out to the renal papilla, gets picked up in a minor calyx, gathered in a major calyx, right, into the renal pelvis, and then on its way out, although you haven't learned these structures yet, let's go ahead and talk about them. It goes ureter, urinary bladder, urethra, toilet. Okay? All right, so eventually you will have to do some kind of trace that involves that. It's really not very hard. This is all the gross anatomy, but we'll put microscopic anatomy together with it before you would have to do a trace. But now what I want to talk about is the blood flow through the kidney. The blood flow through the kidney is not just anatomically important, it's also physiologically important. So I'm going to show it to you in two ways. I'm going to take you through the blood flow in an image and your notes, and then I'm going to show you the animation that shows you the same thing. Okay, so blood flow through the kidney. So let's go from this figure. Of course, all of the blood that's going to the kidney comes from the renal artery, and that probably doesn't surprise anybody. But then we are going to do the gross anatomical blood flow through the kidney, and then there's microscopic blood flow through the kidney. You're going to have to go through this a couple times, so don't be surprised if you don't grasp it the first time. I promise you it's one of the few blood traces that you're actually really, really going to have to like hold on to all the way through phys and then through pathophys. It's a really important one. Okay, so this figure right here takes us through blood flow to the kidney. All right, come here, you little bugger. All right, so first big picture understanding is all of the blood that is in the renal arteries was just in what vessel before that. So blood to the renal arteries comes blank and then renal arteries. So hopefully you guys remember that it's abdominal aorta or descending abdominal aorta. That's what goes there. Okay, and remember, of course, that the renal arteries enter at the hilus or hilum. They receive a ridiculous amount of blood, about 1.2 liters of blood per minute, when you only have about 5 to 6 liters. And then they're going to branch and they're going to form different regions of vessels as they go through. So we're going to do the gross anatomical blood flow, and then we'll go back and pick up the microscopic. So here's all the microscopic stuff right here. We'll come back and get that. So let's just do the gross blood flow, all the stuff that you can see on this figure, and then we'll come back and get this. All right, so we go from the renal artery in, right? And then from the renal artery, we are going to split. These arteries right here are called segmental arteries, and they run through the renal pelvis, right? And then the segmental arteries are going to split, and they are going to go up, around, and snuggle around the sides of each lobe of the kidney, around each pyramid. These are called interlobar arteries, interlobar arteries. So there's one, here's one, so all interlobar arteries. The blood is going through the interlobar arteries this direction. So it's going up that direction, okay? And then from the interlobar arteries, it's going to arch around the base of each pyramid. So the blood is kind of anastomosing that way and that way. And so this vessel right here is called the arcuate artery, arcuate, okay? And then from the arcuate arteries, it's going to give rise to this one that goes up from the arcuates like little bicycle spokes. These arteries right here that you're seeing, those are called interlobular arteries. So a lobule is smaller than a lobe. So I go renal arteries, and then segmental arteries, and then interlobar arteries, arcuate arteries, and then interlobular arteries. Cool? Okay. All right. Now, from there, there's a whole bunch of microscopic circulation that you'll get into. I'm going to come back and pick that up in just a second. So you're going to go into microscopic circulation right here. That gets kind of complex, but we'll be able to do it in just a second. But before we do the microscopic circulation, I just want to show you that mostly the blood supply back from here is very similar, with one little weird exception that I don't understand. I've never heard a good explanation for it. So now, after I do all the microscopic circulation, which I'll tell you about in just a second, then what I am going to do is I am going to deliver blood back, and I'm going to deliver blood back down. So it is going to go, I went up the interlobular artery, and now I'm going to go back down the interlobular vein. And then from the interlobular vein, I'm going to go through this guy right here, which is going to be the arcuate vein. And then down this one right here, which is going to be the interlobar vein. And then here's the part that doesn't make sense to anybody, including me. Shouldn't these be called segmental veins? I think they should, but they are not segmental veins. They don't get a separate name. They're just considered extensions of the interlobar veins, so they're not called segmental veins. They are nothing. And then this is the renal vein, and then it goes back into the inferior vena cava. So once more through the gross anatomical circulation, then I'll add in the microscopic circulation. Okay, so renal artery, check. Okay, segmental artery, right? And then interlobar artery, arcuate artery, interlobular artery. Now we'll do the venous flow back. And the venous flow back, take it from anyone, and it goes interlobular vein, arcuate vein, right? Interlobar vein, this should be called the segmental vein, but it's not. Back to the renal vein, and then back to the inferior vena cava. Cool? So the only thing weird is, of course, there is no segmental vein. Okay, so, and it says it right here, no segmental vein. Okay, so the only thing that we need to do now is go back and pick up this microscopic circulation, and then we'll look at the animation again. So with the microscopic circulation, and this is a really, really physiologically important thing, so I'm going to try to make sure that I can get this through to you. What happens is up here, right at the cortex and also the junction between the medulla and the cortex, what happens there is you actually get kidney capillaries right next to urine producing tubes. And as we've seen all semester, if you get two microscopic sets of tubes close enough to one another, you can have exchange between them. So what we are going to do is kind of like what happened with the placenta with the fetal capillaries and the maternal capillaries that were not connected, but they were close enough for exchange. So the same thing is going to happen in the kidney. What we are going to have is microscopic blood tubes, capillaries, right next to microscopic P tubes. And the P tubes we'll talk about in just a minute. It's called the uriniferous tubule. But then what I can do goes back to what I was talking about at the beginning of this set of notes. What I can do is exchange and figure out what stays in the bloodstream, what gets dumped into the urine, stays in the bloodstream, dumped into the urine. So you have all of these opportunities for exchange right here, because at this location, which is blown up from right here, microscopic blood tubes, shown blood colors, are next to microscopic P tubes, shown in this yellowish color. And that structure right here, where you have the possibility for exchange between blood and urine, is where the kidneys do the cleansing, and it's called a nephron, okay? And you've got about 1 million nephrons per kidney, and they're really where all of the fizz happens. Okay, so before I get into specifically how the blood and the pee do the exchange, let's just go through the blood supply here to make sure we get the blood flow, and then we'll go back and add the pee part later. Okay, so here we go. So let's pick up from where we were. We were saying, okay, so the interlobular artery is what leads to this. So can you orient yourself? So the interlobular artery is like right here, right? And so now I'm seeing this interlobular artery right here. The interlobular artery is what leads to the capillaries of the kidney via this vessel right here. This guy right here is leading from the interlobular artery, and that one right there is called the afferent, as in bringing to arteriole. Arteriole is going to lead to this weird looking ball of capillaries called the glomerulus, right? So I'm going to go through the glomerulus, and then it's going to lead to another arteriole, and this one leads out of the glomerulus, and it's called the efferent arteriole. And then the efferent arteriole, depending on what kind of nephron it is, can lead to one of two things. It can either lead to these guys, which are called the peritubular capillaries. Peritubular capillaries are called peritubular because they're wrapped all around that tube up there. Or, it depends on the type of nephron. I'll show you these in just a second. Or it can lead to these, and these are called the vasa recta. Those are at the same point in circulation. Vasa recta means straight vessels, okay? And wherever you have all of these blood tubes next to all of these P tubes, you get exchanged. So for instance, I am deciding I want to keep all this glucose in the bloodstream, but I want to get rid of that urea into the urine, right? So all of that process is going on as the blood tubes are going by the P tubes, okay? And now what's going to happen after I finish with all of the exchange between blood and urine, now the blood is going to be cleaner. It's going to be slightly lower volume. It's going to definitely have less urea, should have still a whole bunch of nutrients in it. Then what I'm going to do is I'm going to deliver it back into circulation. So, and I'll go through this one more time and then we'll look at it in an animation. So I'm going to deliver it back into circulation via, now it's going to snuggle back behind here from either the peritubular capillaries or the vasa recta, and it's going to dump back into this vessel. And this vessel right here is the interlobular vein. And then from the interlobular, we are going to be on our way out. Okay, so one more time through this, then we'll show the animation. Okay, so blood came into the kidneys from the descending abdominal aorta, and then it went to the renal artery. Check, right? Segmental, interlobar, arcuate, interlobular artery. From the interlobular artery, you go over to microscopic circulation. There's the interlobular artery. Interlobular artery goes afferent arteriole, check, glomerulus, efferent arteriole, and then one of the two. This picture is confusing because it shows both, but you don't generally have both in one nephron. Either it goes peritubular capillaries or vasa recta, and then pretty much you're done cleansing the blood, and you're going to send it back into the interlobular vein, right? And then on its way out, it's going to go arcuate vein, interlobar vein. These are not called segmental, but they should be veins. Back into the renal vein and back into the inferior vena cava. Okay, so there is a cool little thing that makes this guy right here, the glomerulus, really, really high blood pressure and therefore really, really good at filtration. Let's see where they show that picture really well. So notice afferent arteriole going in and efferent arteriole going out. The glomerulus right here is the site of filtration in the body. Filtration is pressure driven. So if you can do anything to bump up the pressure, you can make it really, really good at filtration. And the kidneys are so good at filtration that all of the rest of the capillaries of your body filter about 1 gallon per day, and the kidneys filter about 47 gallons per day. So part of the reason they're so good at that is that if I blow this up right here, I can see, oops, hold on, that, let me get rid of all these hairs real quick. I can see that the diameter of the afferent arteriole going in, right here, and the efferent arteriole going out, they're different diameters. So if you have a big tube going in and then a really porous capillary bed and then a small tube going out, can you see that the pressure and therefore the filtration will be higher in the glomerulus than it generally would be? Okay, big tube going in, really porous, small tube going out, pressure is bumped up. So the blood pressure is higher in the glomerulus than in other capillaries because the efferent arteriole is smaller in diameter than the afferent arteriole. So more resistance and greater blood pressure. Okay, let's see. So the challenge that I gave you guys in the notes, and I want you to write it out to practice it, is to trace the flow of blood from the abdominal aorta through the kidney. Do not go into the urine if I asked you to stay in the bloodstream. Take a red blood cell and take it from the abdominal aorta through the kidneys and then back into the inferior vena cava. Because on a good day, we shouldn't lose any red blood cells into our urine. Okay, so I want you to practice that, and no, you don't need me to check it because you can look at it. I just went through it like 5 times. Okay, so let's look at the rest of the animation that shows you this. All right, so she was just starting at the renal artery, and now let's follow her.
Renal sinus to form segmental arteries, which branch again to form interlobar arteries. Interlobar arteries pass through the renal columns to the cortical medullary junction, where they branch to form arcuate arteries. Arcuate arteries project along the base of renal pyramids to give off interlobular arteries that enter the renal cortex. Finally, each interlobular artery branches to form a series of afferent arterioles, each of which feeds a capillary network called a glomerulus. The glomerulus forms the vascular core of the renal corpuscle and is the initial filtering component of the kidney. The renal corpuscle and its associated ducts form the functional filtration unit of the kidney, the nephron. The efferent arteriole that leaves each glomerulus enters a second capillary network. For nephrons in the renal cortex, a peritubular capillary network forms around the proximal and distal convoluted tubules. From this capillary network, blood leaves the cortex by flowing into interlobular veins, which drain into arcuate and interlobar veins, and finally, the renal vein. Conversely, the efferent arterioles associated with nephrons at the cortical medullary junction enter a second capillary network along the nephron loop, or loop of Henle. These capillaries are known as the vasorecta. From the vasorecta, blood flows sequentially into interlobular, arcuate, and interlobar veins, and finally, the renal vein.
Sorry, that was probably distracting. Kids, dogs, everybody. Okay. Bye, Superfly. All right, so she went through the same thing, and this is a really cool dissection. I would love to be able to do that dissection. Okay, so the only other thing on this part is to talk about innervation of the kidney. How are kidneys innervated? There is actually there are a lot of plexuses that you guys didn't learn about. One of them is actually the renal plexus that actually innervates the kidney, and it enters at the hilum. I don't know if it's shown on this picture, is it? Let's see if it's shown here. No, it's not shown in this picture. So anyway, there are nerves that actually innervate the kidney, and they have both sensory and motor functionality. The motor function that goes to the kidney is primarily sympathetic innervation that controls the diameter of the kidney arterioles. It may also stimulate hormone release. People aren't quite sure yet. And then the parasympathetic innervation does go to the kidney, but nobody knows what it does. There is cranial nerve number 10 that actually goes to the kidneys, but it doesn't seem to matter whether it's hooked up or not hooked up. So that one's kind of interesting. And then there's sensory function from the kidney, visceral sensation, and that your kidneys can hurt. Okay, so want to talk about the microscopic anatomy of the kidney and a little bit about the function of the kidney. So let's go to our friends, the nephrons, in a little bit more detail. Here's Zach Nephron. And then here's a couple of different kinds of nephrons. So these two nephrons are showing the peritubular capillaries versus vasorecta. When a nephron stays up in the cortex mostly, it's called a cortical nephron. And when it's at the junction between the medulla and the medulla and the cortex, it's called a juxtamedullary nephron. And do you see that this cortical one just has peritubular capillaries, and this juxtamedullary one primarily has a vasorecta, a few peritubular capillaries as well? Okay, so these are nephrons. They are microscopic, and there are more than 1 million per kidney. They are where all of the fizz happens at the kidney. And so the basics of urine formation, super important for you to grasp this early, because if you get it before fizz, then the urinary system in fizz is relatively easy. So I drew a really bad picture. I usually draw this on the board, but I'm working on trying to figure out the whiteboard function on the iPad. I'm trying to figure out how to make that work here, but okay, so we got a bad picture here, which I always drew bad pictures for you guys, but now I have to draw worse pictures. Okay, so basically what we have at the kidney, this is not an anatomical drawing, it's like a physiological drawing, is we have blood tubes next to P tubes, right? Okay, wherever you have blood tubes next to P tubes, you can do exchange. And what actually ends up being peed out is what's called excretion, right? You excrete all of that stuff into your urine. But excretion is actually a combination of 3 separate processes. The first process is filtration, and filtration is pressure driven, and it moves anything that will fit through the holes, water, small solutes, even valuable stuff like nutrients and ions, and small nitrogenous water-soluble waste. It will move those from whatever fluid is high pressure, blood, to whatever fluid is low pressure, filtrate. So the site that occurs right here is that high pressure glomerulus, and I just told you one of the reasons that it's so high pressure is that the afferent arteriole is bigger than the efferent arteriole, so it's really effective at filtration. But that's really general. It just moves anything that fits through the holes out from the blood into the urine. It's not called urine yet, it's called filtrate at that point, but it's, if you leave it out there, it will become urine. But that's not specifics, because you lost valuable stuff like glucose, like that's money. So the next step you have to do is you have to do reabsorption. But you've got opportunities to do this, and reabsorption means I'm bringing things back from what would have been pee back into the bloodstream. Reabsorption is selective. I'm going to bring back all of the valuable stuff like amino acids and glucose, and then if I need it, like if I need calcium, I will reabsorb it. If I need sodium, I will reabsorb it. So what I reabsorb is based basically on the needs of the body, and a whole bunch of water. I reabsorb a whole bunch of water. And then the last step is basically moving stuff out, but this time I'm being selective about what I move out. So the first time was kind of everything that fit through the holes, and then I'm like, no, don't dump grandma's china, right? Don't dump a box of money, and I bring that stuff specifically back in. And then the last time what I do is specifically go through the bloodstream and say, do I need 720 cans of corn or molecules of sodium? And if I have too much, I will dump the excess, and I will also dump any waste products that I didn't get the first time. So I do a 3-step process to figure out what ends up in the toilet. And then my blood, after filtration, reabsorption, secretion, and excretion, my blood will go back in ultimately to the renal vein and to the inferior vena cava, but it will be cleaner. We will have lost some volume because you always lose some fluid. Hopefully we will have kept all of our red blood cells, all of our nutrients like glucose and amino acids. On a good day, you should keep those. But we will have gotten rid of waste products and excesses. So this process right here is all going on in these super curved tubes called the nephron. And it can go on any location where you have blood tubes and P tubes next to one another. Okay, so, and I'll link this ugly picture in your notes. Okay, so filtration, reabsorption, and secretion. These are the basics of urine formation. It's a 3-step process for you to get your blood cleaned. So now let's go back to the nephron. The nephron consists of 2 major components. We already went through the first component of the nephron, which was the vascular component, meaning the blood tubes. And the vascular component of the nephron, we're going to go through it again one more time, starts with the afferent arteriole, okay, the afferent arteriole right here, glomerulus, efferent arteriole, peritubular capillaries or vasorecta, and then it dumps back into the macrocirculation when it dumps back into the interlobular vein. So this is all the microcirculation of the kidney, the vascular component of the kidney. But you can't do exchange between blood tubes and P tubes unless you also know the P tubes. And here's the mistake that everybody makes, and I don't want you to make. If I ask you to do a trace of blood, like with a red blood cell, you go in through a blood vessel and stay in the blood, right? You don't end up peeing out red blood cells. But what if I asked you to track a molecule of urea, which is a nitrogenous waste product, until it hits the toilet? Well, then we're going to start in the blood and we're going to end up in the urine. Okay, so we'll practice that as we go through as well. So this whole thing, the P tubes, they begin as this blind end sac right here that is around the glomerulus, and this is called either Bowman's capsule or the glomerular capsule. But important, you cannot use the terms glomerulus and glomerular capsule interchangeably because they don't mean the same thing. So let me show you what you're actually looking at if you blow this up really, really far. Okay, so we already looked at the afferent arteriole and the efferent arteriole and the glomerulus. But the glomerulus is a capillary, so why does it look so weird in this picture? Because capillaries are not made of those cells. They're made of simple squamous epithelial cells, and those are some weird-ass looking cells. So what am I looking at here? What I'm looking at here is the glomerulus. Imagine that the glomerular capillaries are my fingers, right? But the capsule works like a softly blown up balloon that you push onto the glomerulus so that the balloon coats every single capillary like a glove, and then there's a space, and then there's another layer of the balloon. So you basically get a visceral layer of Bowman's capsule that you're seeing right here with these weird looking cells called podocyte cells, podo as in foot. Here's one right here. And that is what is on top of the actual capillary, okay? And then you get a space, right, before you get to the parietal layer, right? And then what is between the visceral and the parietal layer is any fluid and solutes that you push out from the capillaries, right? They get pushed through, it's a fenestrated capillary, they get pushed through the fenestrated capillary. And as they go through the fenestrated capillary, they slip through these little feet, little spaces between the podocytes, and then they get caught in this space right here, the space between the visceral and the parietal layer of Bowman's capsule or the glomerular capsule. So the capsule is the first place that you have P. It's called filtrate until it hits another structure, but first place that you have anything that could possibly become urine. And this is a cool figure that actually shows you a scanning electron micrograph of that. And then this is a colorized scanning electron micrograph, which is super cool. So what you're looking at here is the podocyte cell and these filtration slits between the feet. So you're looking right here at the visceral layer of Bowman's capsule, and Bowman's capsule is also called the glomerular capsule, but it's not called the glomerulus. Okay, all right. So where is this relative to the gross anatomy? Generally speaking, this structure right here is going to be located in the cortex. Could be higher or lower in the cortex. And so you got the visceral and the parietal layer, and with a space between it. And so what you call these things is, where are we? I lost my place. Okay, so this structure right here, the capsule plus the glomerulus, is called the renal corpuscle. And the renal corpuscle is what you're seeing right here. So the histology of the renal corpuscle. So the renal corpuscle has both the capillaries and the capsule. It's the first place where you have exchange between blood and urine. It is the primary site of filtration, and it's really good at filtration for the pressure reason that we talked about, because the afferent arteriole is bigger than the efferent arteriole, but also because it's really porous, right? Lots and lots of holes. And also because there's over 1 million of them per kidney. So you end up with a lot of surface area, really, really permeable and high pressure, which makes you fantastic for filtration. Okay, so let's go back to our nephrons for a second. All right, so let's say, oops, that's not what I wanted to do. Let's say that a molecule of urea got pushed from the blood. By the way, urea is a byproduct of protein breakdown, mostly. And you always are going to have it because you have to have proteins in your diet. But you need to get rid of it or else it's going to screw up a bunch of things, among them your pH. So you got to get rid of it. So now what we're going to do is we're going to follow it out. So let's say it got pushed into the capsule, from the glomerulus into the capsule. Then it's going to make its way out, and it's going to make its way out by first this curvy tube that's right next to the glomerulus. That curvy tube that's right next to the glomerulus is called the proximal convoluted tubule, often abbreviated PCT, and it has pre-P in it, filtrate, okay? Does not have blood in it. If it does, you are having a very, very bad day. Proximal means close to the glomerulus. Convoluted means curved, and tubule means little tube. Now, way back at the beginning of the semester, you learned that kidney tubules are made of simple cuboidal epithelium. You didn't have any idea what we were talking about, but we were talking about these tubes. Proximal convoluted tubule and the distal convoluted tubule are mostly made of simple cuboidal epithelium. I'll show you what that looks like. Not that I won't hold you responsible for this, but look, these are the cells right here that you saw all the way back at the beginning of the semester, right? And if you do a cross section, you will see, you will see some simple cuboidal epithelium right next to some capillaries, because what you are looking at is all of those blood tubes next to the P tubes. Blood tubes being simple squamous epithelium and the P tubes being simple cuboidal epithelium. All right, so now what we are going to do is we're going to keep making our way out toward the toilet. Ready? So I push that molecule of urea into the proximal convoluted tubule. And then if you can follow it, it's hard to follow in this one. The proximal convoluted tubule will lead to the, oh, there it goes. It will lead to the descending limb of the nephron loop, also called loop of Henle, and then up to the ascending limb of the nephron loop. And then it will lead to this distal convoluted tubule, which if you follow it really well, will dump right back into this guy right here, which is the collecting duct. So let's look at it in the notes, and then we'll look at the animation too. So the proximal convoluted tubule leads to the loop of Henle, also called the nephron loop, and it goes descending and then ascending. The loop is mostly about water reabsorption. Desert animals have really long loops so that they have a lot of opportunity for water reabsorption. And then it goes to the distal convoluted tubule, and the distal convoluted tubule is the end of the nephron proper. This guy right here is called the collecting duct, and the collecting duct actually collects from multiple nephrons, like this one, and there was one there, one here, one here, one here, one here. So the collecting duct is actually not part of any specific nephron. It's actually gathering from multiple nephrons. So the collecting ducts receive the filtrate urine from several nephrons, and the collecting ducts are the reason that, so if I followed all of this and it doesn't show the microscopic structures, what would happen is the whole nephron portion would start up here, and then the collecting ducts would collect the urine as they're going down. And do you see how these look like almost parallel, not quite, but they're all basically pointing toward the renal papilla? That's because what you're seeing is the direction that all these collecting ducts are going. So the collecting ducts are going to dump. They're going to go through the renal pyramids. They give them that kind of striated appearance, and all of the collecting ducts are going to merge, and they're going to dump the urine and that urea that I took out of the bloodstream into, they're going to dump it from the renal, from the renal papilla, right? into the minor calyx, the major calyx, the renal pelvis, the ureter, the urinary bladder, and then the urethra. So I just challenged you right here to trace the flow of filtrate and later urine from its production until it enters the ureter. So what I want you to think about is that molecule of urea that was in my bloodstream, and I needed to get it into the toilet. I will give you a hint. The place that it goes from blood to urine should be from the glomerulus to the capsule. And then let's say it doesn't ever go back into the bloodstream. So it's going to follow its way out through the capsule. Capsule, proximal convoluted tubule, loop, distal convoluted tubule, collecting duct, renal papilla, minor calyx, major calyx, renal pelvis, ureter, urinary bladder, urethra. Okay, so let's watch the animation that shows you the microscopic circulation of the kidney. So this basically follows along immediately after that one. Okay, get rid of all my annoying arrows. Get out of there, you little buggers. Okay.
The functional filtration unit of the kidney is a microscopic structure called the nephron. Each kidney contains approximately 1.3 million nephrons. Each nephron consists of a renal corpuscle and a renal tubule. The renal corpuscle is a bulbous structure in the renal cortex, consisting of a capillary network called the glomerulus and a surrounding glomerular capsule, also known as Bowman's capsule. Blood vessels enter and exit the renal corpuscle at its vascular pole. The tubular pole is continuous with the renal tubule. The glomerular capsule has two layers. The visceral layer envelops the capillaries of the glomerulus. It is composed of specialized epithelial cells called podocytes, which have multiple interdigitating foot-like processes called pedicels. The parietal layer is a simple squamous epithelium that forms a capsule around the glomerulus. The capsular space lies between these two layers. Water and some solutes of blood plasma pass through a filtration membrane formed by three components of the glomerulus. Small pores in the capillary endothelium called fenestrations, a basement membrane between the endothelium and the podocytes, and narrow spaces called filtration slits between pedicels. The renal tubule is composed of a proximal convoluted tubule, a nephron loop, and a distal convoluted tubule. The extensively coiled proximal convoluted tubule originates at the tubular pole of the renal corpuscle and is continuous with the nephron loop. Each U-shaped nephron loop, also known as the loop of Henle, consists of a descending limb that extends into the medulla and an ascending limb that returns to the cortex. The nephron loop is divided into thick and thin segments. The thick segments are lined with simple cuboidal epithelium, and the thin segments are lined with simple squamous epithelium. Beyond the ascending limb of the nephron loop, the renal tubule coils again to form the distal convoluted tubule. Several distal convoluted tubules drain into collecting ducts that pass through the medulla of the kidney. These merge to form papillary ducts that drain into a minor calyx. As fluid passes through the renal tubule, useful solutes move out of the tubular fluid and return to the blood, and waste products in the blood move into the tubular fluid, ultimately to be excreted in the urine. Two types of nephrons can be identified based on their location.
Okay, so gave you a visual of the same thing is what we were going for. All right, so I would recommend that you color code in this thing. Oh, why did it start at 4? I'll fix that for you. It should start at 1. So I'll fix it for you. I don't know why I did that. I'll fix it for you. So I would recommend that you go through and label and color code all of the blood tubes red I mean, I know they'll do gas exchange, but you can color them all red so that you can keep track of where a blood cells should stay. And all of the P tubes yellowish so that you can keep track of where the urine should go. And then make sure that you can label and identify all of those, and I'll fix it on your Google Docs notes in just a second. So I'm going to stop there for today, and then I will put up another one of these early next week. You have a vessels one. I don't know how many of you guys have watched. And then we will do the second urinary system, and we'll start on reproductive system next week. I will see you guys soon.